So good morning, everyone. And I'd like to thank the organizers for the invitation. And I'm very excited to be here and tell you all about my experiences with living on the edge of salts and liquids and what we found out about the selective hydrogel at the heart of the nuclear pore. So since this project was very collaborative, I'd first and foremost like to thank all the people who worked on it. John, first of all, who did all the non-NMR work, so protein expression, biophysical methods. He works in Professor de Gerlich's group. And I'd like to thank my PIs, Lauren Andreas and Professor Christian Griesinger as well. So first, a short introduction to the system we were working with. The hallmark of every eukaryotic cell is its cell nucleus, which is separated from the cytoplasm by a double membrane called the nuclear envelope. Of course, this nuclear envelope can't be impermeable since material, including RNA, nuclear proteins, have to flow somehow through it. And what takes care of this is the nuclear pore complex, which are 1 megadalton large complexes scattered across the nuclear envelope's membrane. So the most important property of these complexes is that they act as a selective sieve, which means that they let small molecules diffuse through freely. However, at larger sizes, this diffusion becomes increasingly restrictive, and the cutoff limit is around 30 kilodaltons, unless the cargo molecule is coupled to a protein called the nuclear transport receptor, which then facilitates its diffusion by up to 5 orders of magnitude. So with this, we can see very nicely here on these microscopy images, if we incubate cell nuclei with the inert protein M-cherry, 28 kilodaltons in size, we can see that the protein is excluded from the cell nuclei. However, if we incubate it with the NTR, nuclear transport receptor NTF2, then it can diffuse nicely into the cell nuclei. The proteins responsible for this selective behavior are called FG nucleoporins. These are anchored to the inner scaffold of the nuclear pore complex, or the MPC core. And their most crucial part is their low complexity FG domains that get their name from the fact that they are very rich in phenylalanine glycine motifs. So up to 15 FG motifs can be found in a single FG domain. These FG repeats occur at a high concentration inside the nuclear pore, millimolar concentration, and their most important property is that they phase separate or are cohesive. What this means is that if we purify the protein under denaturing conditions and then dilute it with a physiologically relevant buffer, we obtain these nice little droplets or particles, whatever we choose to call them, that actually recapitulate the selectivity properties of the cell nuclei, as we saw on the previous slide. A way to characterize this phase separation behavior cohesiveness is by something we call centrifugation assay. It's very simple, and I'll be referring to this later on, so I'll introduce it quickly here. Basically, if we prepare these self-assembled FG-hydrogel particles and we centrifuge them, we obtain both a pellet and a supernatant. And if we run them on an SDS gel, we can see that in case of a phase separating protein, most of the protein will be found in the pellet, whereas a little bit in the supernatant. And from this behavior, we can also or from this SDS gel, we can also then infer the critical concentration from the relative band intensities. So it was long debated what the basis for the selectivity of the nuclear pore complex is, and a relatively widespread and well-accepted model was the selective phase model, which says or postulates that the FG motifs in these FG domains come together to form sort of a selective mesh. And whenever an NTR comes, it locally can melt this mesh, which then after the nuclear transport receptor has passed through with its cargo, the mesh can reform. And indeed, mutagenesis studies show that phenylalanine is required for phase separation. So if it is replaced, phenylalanines are replaced with serines, then the cohesive behavior is lost. However, it was unclear whether it is polyvalent aromatic contacts that are important for phase separation or hydrophobicity. And further, biophysical characterization of the hydrogel resonance assignment was hampered by the sequence's degeneracy. So our collaborators took a very nice approach to this, and they simplified the wild type sequence, which is about 600 residues long in total, to a perfectly repetitive sequence that as you can see, reflects the selectivity properties of the wild type sequence basically perfectly, as well as the phase separation properties, so the critical concentration. What they did was basically they found that these FG motifs occur roughly every 12 residues in the wild type sequence. So they introduced these every 12 residues in the model sequence as well. And then they did a statistical analysis of the amino acids occurring in the spacers of the wild type sequence and designed spacers to reflect this. So this was the sequence that we used for our studies. We embarked on our adventure asking 4 questions. We wanted to find out whether on a molecular scale, the FG phase behaves more like a solid or a liquid. We wanted to see whether the peptide chain has a secondary or tertiary structure. We wanted to see which residues are involved in phase separation, and we wanted to find out more about the thermodynamics of phase separation as well. I will depict these 4 questions with these 4 little icons, and you'll see them on my slides whenever I'm referring to one of these questions. So first of all, we had to pack the sample. This was much easier said than done. It's a very, very viscous material. I can't really say whether it's solid liquid. We did a FRAP fluorescence recovery experiment. We saw that the diffusion is very slow. So we decided to go for solid state NMR. We're solid state NMR spectroscopist anyway in my group. So we somehow got it into the rotor after much struggle. And we could record our first spectrum, both the wild type sequence and the perfect repetitive sequence. So what we saw was, first of all, narrow lines, even at low magic angle spinning frequencies and low power proton decoupling. And we also saw that our cross polarization intensities were much lower than the direct polarization intensities, which all points to the fact that probably on a local scale, this gel is very mobile. I would like to point out the fact that usually we think of cross polarization signal as signal belonging to the rigid regions in the protein. However, we suspect that in our case, since the transfer times are very long, above 10 milliseconds, this actually stemmed from the cross-polarization transfer happening through weak or J couplings. And this has actually been shown in literature before that this can also happen. So then we thought, okay, why not try it in solution NMR probes? Since there are a lot of established sequences, it would just be much more convenient. So first we pipetted the dispersion of these little particles into an NMR tube, but these settled, stuck to the wall. Our spectra was gone in half an hour. Then we thought, okay, we might as well try rehydrating the lyophilized protein. So we stuffed that into a solution NMR tube and tried hydrating it. However, there were little pockets of buffer and even worse air remaining in there, which was really detrimental to our spectrum. So this didn't work either. So finally, we had to revert back to solid state NMR. And this worked really well. So we could pack the rotor and by centrifugation rehydrate this lyophilized protein. And even with slow magic angle spinning, we could spin out all the magnetic susceptibility and homogeneities stemming from these little buffer pockets. However, this meant that we also had to adapt some of our solid state NMR pulse sequences for use on solution-like samples. One of the most important things is that in solid state, we don't have any gradients to work with. So, for example, here is the simple HNH or HSQC correlation spectrum that I used. It's very similar, of course, to that used in solution state. One thing you have to pay attention to, though, is that here we can't use the out and back transfer. We have to go all the way to in-phase magnetization, because if we're using the Mississippi water suppression, usually used in solid state, that would scramble the magnetization if there were any magnetization remaining on protons. Of course, we're using NF transfers. We got pretty lucky with our TROSY sequences there. We just had to take out the gradients and they worked as they were. That was pretty much a miracle. And then for 3D experiments, assignment experiments, again, we're using NF transfers. There are well-established sequences that work even without the use of gradients. But again, here we had to, so here we had some problems with artifacts, which we could get rid of only by adding some Z filters, which I've highlighted here in blue. And we also put in some water suppression. Why not? Helps with the spectrum quality. So after this, we could record our first 2 and 3D spectrum on the sample. both the wild type protein and the perfectly repetitive sequence. So what we see is if we overlay the wild type spectrum shown in dark blue and this light green perfectly repetitive spectrum, they overlay very well. Both have a pretty narrow line width, at least for the isolated peaks. Okay. Yes. And oops. So this kind of indicates that they have similar properties. They're probably both IDPs. So again, this is a fully proteinated sample at low magic angle spinning rates. However, since this sequence is 600 residues long, we can see that assignment is pretty much impossible. So we had to go for the perfectly repetitive sequence to assign that. And as you can see there, we have very nice narrow lines, well isolated from each other. So they're those we could assign. One thing I'd like to point out is that we have 11 non-proline residues showing up here. So 11 peaks, which correspond to the 11 non-proline residues. So this also means that all our repeats are equivalent. And again, this low chemical shift dispersion indicates that we're talking about an IDP. So we've kind of answered our first two questions. Our sample behaves kind of like a liquid on a local scale, and there's no secondary structure. We also took a look at the HC correlation spectrum. Again, nice narrow lines. The aliphatic and aromatic regions are separated. If we zoom in on the aromatic region for the phenylalanine, we can see 3 different peaks, which correspond at 221 intensities, which correspond to the 3 different HC groups in the phenylalanines side chain. So this also shows that all the phenylalanines are also equivalent in the gel. There are, at least on the NMR time scale, there is no secondary or tertiary structure of phenylalanines packing into some high order structure. Okay, so next up, we wanted to understand a bit more about the phase separation. So we played around a bit with the gel. First, we checked its temperature dependence, and we saw, which was actually quite surprising, that if we heat up the protein solution, phase separation occurs. This is quite unusual. So there are very few proteins that exhibit this behavior, and it's called lower critical solution temperature, or LCST behavior. And this is reversible. We also took a look at this with the centrifugation assay, so that if we saw that, if we heat up our protein, then less and less protein is left in the supernatant, and more goes to the pellet. And to quantify this a bit more, we could do dynamic light scattering experiments, where we saw that if we heat up our sample, all of a sudden there's a very strong increase in particle size, which indicates phase separation happening. So with this, we could record saturation, concentration, temperature pairs and construct a phase diagram, which I will get back to later. Okay, so we also wanted to see what the dynamics of inside this hydrogel are. We already know that it's liquid-like, but let's take a closer look. So for this, we use the tracked experiment, which is TROSY for rotational correlation times. And it's a very nice and pretty easy to record experiment. It is based on the fact that we record the transverse relaxation rates of nitrogen in both its alpha and beta state. And if we look at these expressions, we can see that these two are determined by the same 4 parameters or factors. However, the transverse cross-correlated relaxation rate appears with an opposite sign. So if we subtract the 2 from each other, we are left with only the transverse cross-correlated relaxation rate. And we have, for example, eliminated the contribution of chemical exchange. Now this eta can be expressed as following. None of it matters what is what. What is only important is that all these are known parameters inside this equation. And we only have one unknown, which is the tau c. So if we measure our beta and our alpha, we can calculate tau c for each residue. And here is the pulse sequence that we use. So this is basically just a TROSY sequence with a relaxation delay in there that has a pulse to refocus chemical shift evolution during the relaxation delay. And to record the beta sequence, we just basically have to flip the proton ionization, and then we select the other components of the TROSY peak. So with this, we could measure our rotational correlation times in both the solution state and the solid, or the gel phase. So we see that in both cases, it's nanosecond time scale mobility. In the gel, it's a bit more rigid. Oopsie. However, we see that in the gel, there is a very distinct maximum around phenylalanine, meaning that probably the phenylalanines interact with each other. In solution, we don't see any such profile, but we were interested whether there could possibly be intramolecular interactions present even in solution. So we prepared a shorter peptide, which instead of 52 perfect repeats as our full-length perfectly repetitive sequence, this only has 7 perfect repeats. And indeed, we saw a 2-fold decrease in rotational correlation times, which confirmed that even in solution, there are intramolecular interactions present. And that slowed down the motion of the protein chain. Then we thought, okay, let's try to change the temperature. We saw some interesting temperature effects before. So we heated up our 7X protein as a control, and we saw that, no big surprise, it became more mobile at higher temperatures. However, when we heated up our gel, we saw something very different. It actually became more rigid, and the profile also changed. So we can see that there are now 2 maxima. One is around proline. This is no big surprise since proline has a rigid 5-membered ring in the peptide chain. However, we see a very distinct maximum around leucine now, and leucine's rotational correlation time is actually higher than that of phenylalanine. So this suggested to us that there might be something more going on there. So we recorded a carbon filtered nosy experiment, so HHCH basically. And while this experiment does not show us contacts between identical residue types, since those are on the diagonal, it does show us contacts between different residues. And indeed, we could observe contacts from phenylalanine to a whole bunch of hydrophobic side chains, so CH3 and CH2 groups. However, something that was not seen before was we could also observe purely hydrophobic contacts within the hydrogel. So to play around with this hydrophobicity a bit more, John did a few mutagenesis experiments. So first, kind of as a control, he mutated away phenylalanines to alanines. This had already been done before with mutating phenylalanines to serines, and no big surprise, the protein did not phase separate anymore. However, we saw that when we mutate leucines to alanines, the same thing happens. Phase separation is eliminated. But now we kind of had a hunch that, okay, it might be the hydrophobicity that is important. So we mutated leucine to the slightly less hydrophobic valine. And indeed, phase separation was preserved, but our critical concentration became significantly higher. So we thought, okay, phenylalanine is hydrophobic, leucine is hydrophobic. Why not just mutate phenylalanine to leucine? Well, that doesn't work. Phase separation is gone. However, if we think about it, phenylalanine has more hydrophobic carbon atoms than does leucine. So we decided to replace 1 phenylalanine with 2 leucines. And this leucine-enriched sequence actually again showed phase separation and a submicromolar critical concentration. So this again, this confirmed what we had been thinking, that it is hydrophobic contacts or hydrophobic interactions that play a really big role in phase separation, not necessarily just these aromatic contacts between phenylalanines. So then we know that hydrophobic interactions are very sensitive to salt. So we played around a bit with the salt concentration. We saw that if we increase salt concentration, more and more proteins goes to the pellet, and yeah, the critical concentration drops. And if we look at a FRAP experiment, again, we see that the already slow diffusion, if we increase the salt concentration, slows down even more. So the recovery becomes slower. And if we look at the dynamics in the hydrogel and function of the salt concentration, we see that with increasing salt concentration, the protein chain becomes more and more rigid. And not only is this true in the gel, but also in solution, which was actually a little even surprising. This even works for both for the full-length sequence and the shorter sequence, this 7X repeat. So if we increase the salt concentration, these chains become more rigid, again, pointing to hydrophobic interactions. And finally, to quantify the thermodynamics of phase separation, we used our phase diagrams that we constructed from our DLS data. And using Van 't Hoff's equation, these very basic thermodynamics equations, we could calculate by fitting the phase diagrams, we could calculate the enthalpy and entropy of phase separation. And we found both to be positive. So basically, in order for delta G to be negative, this term has to be large. So this phase separation is driven by entropy. Okay, so as a conclusion, we found that the hydrogel, even in its gel form, exhibits high local mobility, and we could observe transient contacts uniquely by NMR. We found that the hydrophobic residues are more rigid and their interactions are salt dependent. We could see that total sequence hydrophobicity determines phase separation behavior, and we found that our phase separation is driven by entropy, so it's a hydrophobic phase separation. And finally, before I finish, I would like to advertise something that actually becomes important for solid-state NMR spectroscopists if they are working, especially with proton detection, where we have very nice narrow lines on protons, sometimes around 0.1 ppm. So the thing is, if we measure long experiments, the magnetic field drift during our experiments can become comparable with our line widths, which is of course very detrimental to the spectrum. We don't have a lock in solid-state NMR. So we need to somehow compensate for this. One thing that we can do is we can split the experiment up into shorter experiments and set the magnetic field back between each block. However, sometimes the experiments end at night or over the weekend. It's not very fun. And also, this method does not compensate for the field drift that happens during one block. However, we found a way around this, and we wrote a script for this. And this basically applies a linear drift compensation. So what you have to do is you, again, split the experiment up into shorter blocks and measure the magnetic field with a reference signal. So we just record a 1D spectrum, and we use the water signal for this purpose. And we implement a linear phase ramp on the FID. And so, sorry, so during all these blocks, we can approximate the magnetic field drift as linear, roughly. So what we do is we can then correct each block using a linear phase ramp. What this does, so we apply this to the FIDs, and after Fourier transformation, this results in a frequency shift. And we can do this for both direct and indirect dimensions. And I'd like to show you what effect this has. So for example, here's an H&H spectrum recorded that was recorded for long and had a large frequency drift. So from these butterfly peak shapes, we can obtain these nice round peak shapes after drift correction. And it's even more significant for 2 and 3 and 4D experiments, which can be sometimes a week long. So here's an HNCO experiment that I recorded for about a week. And you can see that without drift correction, we have a much lower signal to noise than with drift correction. So this script can be run directly from Topspin. You only have to give it 3 parameters: the starting shift of water, so the frequency before and after. So basically how much it has to shift, and then your experiment number. And you can use it for 2, 3, or 4D data, and on planes, points, news data. So we wrote it for all kinds of data. Now we have it for both Topspin version 3 and 4. So you can contact us for the script or an older version. So it's for Topspin 3 version of the script we also have in our publication. And with this, I'd like to thank you for your attention. And if you're interested in any more details about the nuclear pore project, we have recently published a paper, so you can probably find it there. Okay, thanks.